In an earlier chapter, we learned about the basic structure of DNA. Today, we'll look at some important experiments that allowed biologists to understand what that DNA was, in fact, the hereditary material, and later to understand its structure. Lastly, we'll look at how DNA serves as a template for making copies of itself. At about the time that Mendel's work on heredity was rediscovered at the turn of the 20th century, biologists were starting to understand the link between Mendel's experiments and chromosomes they saw under the microscope. Unfortunately, they weren't really sure that what those chromosomes were made of. Many people proposed that proteins were the more likely hereditary material, because proteins are very complex with 20 different amino acids. DNA, on the other hand, seemed less likely because it was simpler, with only 4 nitrogenous bases. Scientists at the time didn't understand how such a simple molecule could hold such promise for encoding the complex data required for driving heredity. In 1928, Griffith did a series of experiments that showed that a genetic trait can be transferred between individual organisms. He started off with 2 different strains of bacteria. One strain had a smooth coat and was pathogenic. The other was rough and was non-pathogenic. Because of this difference, when the pathogenic strain was injected into mice, the mice died. When the non-pathogenic strain was injected, the mouse survived. Not surprisingly, if he heated the pathogenic cells to kill them, they could no longer kill the mice when injected. However, if he mixed together heat-killed pathogenic cells with the non-pathogenic rough cells, the result was pathogenic smooth cells that were capable of killing mice. So notice, neither of these 2 ingredients alone could kill the mice, but when they were combined, the non-pathogenic cells, the only living cells, were transformed into pathogenic cells. Griffith concluded that something in the pathogenic cell was able to be transferred out and into the non-pathogenic cell to transform its phenotype from non-pathogenic to pathogenic. Furthermore, whatever was transforming the cells was able to be inherited. In the early 1950s, Hershey and Chase did an experiment to try to determine whether the genetic material was protein or DNA. They used a virus called a bacteriophage, or phage for short, that infects bacterial cells. Usually, the phage lands on the surface of the bacteria and injects its genetic material into the bacteria. Some viruses, like coronavirus, use RNA as genetic material, and others have DNA. In any case, the genetic material enters the bacterial cell and uses the bacterial cell's protein manufacturing machinery in order to make many copies of each of the components of the virus. Eventually, these pieces are assembled and they lyse from the cell, killing the bacteria and going on to infect more bacterial cells. For this experiment, they made radioactive DNA by labeling the DNA with radioactive phosphorus, and they made radioactive protein by labeling amino acids with radioactive sulfur. In their experiment, Hershey and Chase grew bacteriophage to include the radioactive DNA and the radioactive protein. Their idea was that the genetic material was inserted by the bacteriophage into the bacteria. So if the genetic material was DNA, then they would see radioactive DNA in the bacteria. If, on the other hand, the radioactive material was protein, then radioactive protein would be found in the cells. So in their experiment, they used radioactive bacteriophages to infect bacteria, and then they put the mixture in a blender and blended it so that the virus particles were sheared off. After the virus was separated from the bacteria, they used a centrifuge to spin the material. The bacterial cells are heavier and denser, and they form a pellet at the bottom of the centrifuge tube. The viral particles are less dense and remain suspended in the water and form the supernatant. They then tested the pellet, and the supernatant for radioactivity. What they found was that when the protein was radiolabeled, the supernatant was radioactive, indicating that the protein had stayed out of the bacterial cells. However, when the DNA was labeled, the radioactivity appeared inside the cells, indicating that the bacteriophage had injected DNA into the bacteria. This indicated to them that the genetic material was, in fact, DNA. In the 1940s, Erwin Chargaff did a series of experiments isolating DNA from different species. He found that species varied in their nitrogenous base composition. It made sense that the genetic material, which gives different species their differences, would have different composition. And Chargaff's work suggested that it was, in fact, DNA. He also noticed that the amount of adenine was always approximately equal to the amount of thymine, and the amount of cytosine was always similar to the amount of guanine. These became known as Chargaff's rules and influenced Watson and Crick when they later figured out the structure of DNA. We can use Chargaff's rules to estimate the amount of any base if we know the content of any other base. For example, the bacteria Burkholderia pseudomallei has a very large amounts of guanine. If this bacteria has 34% guanine, then what percent of its DNA is adenine? Well, if 34% is guanine, then according to Chargaff, 34% should also be cytosine, giving us a GC content of 68%. The remaining 32% must be adenine and thymine, and those must be in equal amounts. So 16% of the genome is adenine. In 1953, Watson and Crick figured out the molecular structure of DNA. They realized that the molecule fits together very well if it was in the shape of a double helix, with nitrogenous bases facing into the interior of the helix. This structure also matched up very closely with Chargaff's rules and a lot of other evidence. Before we look at its molecular structure in more detail, there are a few important things to note. First, note that the DNA has two basic parts: its backbone, here represented by the ribbon in blue, and its nitrogenous bases, which face each other and branch off the backbone. Next, what do we mean by a double helix? There are in fact two molecules, each with a backbone and nitrogenous bases. Each of these molecules is a single helix, but DNA exists as a double helix with the two molecules wound around each other. The third thing to notice is that these two molecules are anti-parallel. They're parallel with each other in the conventional sense. They go in the same direction. But we say that they're anti-parallel because their orientation is opposite. One molecule is oriented up and the other is down. Okay, so let's review the structure of DNA in a bit more detail. Remember back to a previous presentation where we showed that a single nucleotide consists of a ribose sugar with 5 members. On the 5' end of that sugar, there's a phosphate group, and on the 1' carbon, there's a nitrogenous base. Notice that the 2' carbon has no hydroxyl group, and so this is deoxyribose sugar. RNA, ribonucleic acid, has a hydroxyl group on the number 2 carbon. Remember also that nucleotides polymerize by adding new nucleotides to the 3' carbon on the sugar. As a result, we always say that DNA grows or polymerizes at the 3' end. We can now see the backbone that forms the helix of the DNA. The backbone consists of alternating sugar and phosphate groups, and for this reason, we call it the sugar-phosphate backbone. You can see here that the 4 different bases that exist in DNA: thymine, guanine, cytosine, and adenine. Remember that guanine and adenine are purines and are larger with 2 ring structures. Thymine and cytosine are pyrimidines and are smaller with a single ring. If you need a refresher on this, please look back at the video on nucleic acids. Because of the size differences between purines and pyrimidines, a purine always pairs with a pyrimidine. Two purines together would be wider than the observed diameter of the double helix, and two pyrimidines would cause the double helix to be narrower than what's observed. The pyrimidine thymine and the purine adenine have two atoms that face each other that can hydrogen bond with each other. Guanine and cytosine have three atoms that can hydrogen bond, and so the hydrogen bonding that forms between these nitrogenous bases is stronger when a guanine is paired with a cytosine compared to adenine pairing with thymine. However, notice that adenine always pairs with thymine and guanine always pairs with cytosine. This strict pairing is what Chargaff noticed when he found that the amount of adenine is always equal to the amount of thymine. Here's another view of what the two molecules look like when they hydrogen bond together. You can see a single DNA nucleotide with the sugar phosphate backbone, and you can see that each molecule has a 5' and a 3' end. Remember that these are designated by the counting of atoms on the sugar molecule. Notice that the anti-parallel strands are running in opposite direction, with the 5' at the bottom of the page and the 3' at the top. The nitrogenous bases hydrogen bond with each other such that thymine always bonds with adenine and guanine with cytosine. Again, this pairing of a purine with a pyrimidine ensures that the spacing of the molecule is constant. Finally, and perhaps the most important thing to remember, is that DNA is a way of storing information. The information is stored as a sequence of nitrogenous bases. The sugar phosphate backbone is constant through its length. It's the nitrogenous bases that form the code of DNA. We'll see how that code is used in order to generate proteins later. One of the observations that Watson and Crick had made was that the base pairing of the nucleotides suggested that there could be a mechanism for replicating DNA using one strand as a template and the other strand being built upon that template. Michelson and Stahl did a series of experiments to determine exactly how this replication occurs. They tested three different hypothetical mechanisms for DNA replication. In the conservative model, one double helix would serve as the template to create a second double helix, but these helices would be separate. In a second model, called the semiconservative model, each side of the helix would serve as a template to create another molecule. The new molecule would be interwoven with the original molecule. In the third model, called dispersive, arbitrary chunks of DNA would serve as templates, and the resulting molecule would be a combination of all of these separate chunks. In Michelson and Stahl's experiment, they grew bacteria in a heavy isotope of nitrogen. The result of this was that all of the DNA incorporated into the bacterial genome was made up of this heavy nitrogen. During the experiment, they transferred the bacteria to a lighter isotope of nitrogen and allowed the bacteria to incorporate that lighter nitrogen into its genome through either one or two rounds of DNA replication. After each round of replication, they took a sample of the mixture and centrifuged it. The centrifugation separated molecules that had incorporated the light isotope from the rest of the DNA that had the heavy isotope. The location of the radioactive bands within the centrifugal column could be identified. If the conservative replication model was correct, then after one round of replication, half of the molecules should have contained the heavy isotope and half of the molecules should have had the new, lighter isotope. After a second round of replication, the same heavy molecule should have given rise to another light molecule, and the light molecule should have given rise to yet another light molecule, resulting in 3 times as many light molecules as heavy molecules. In the semi-conservative model, the heavy molecule and the light molecule remained intertwined with one another, and as a result would centrifuge to an intermediate position. After the second round of replication, half of the molecules should be composed of only light nitrogen, and the other half of the molecules should be composed of half light and half heavy nitrogen. In the dispersive model, the first round of replication would result in molecules that are half heavy and half light. The second round of replication, however, should result in molecules that are about 1/4 heavy and 3/4 light. The results from the experiment showed that after one round of replication, all of the molecules were composed of half heavy and half light nitrogen. This eliminated the conservative model. After a second round of replication, Michelson and Stahl found that there were bands indicating some light molecules and some of molecules that were a mixture of the light and heavy nitrogen. This suggested that the semi-conservative model was correct. So, one molecule of the DNA double helix is able to serve as a template for making a new molecule. In this picture, you can see that the lower molecule is the template and the new strand is being assembled above it. The end of the new strand is, of course, the 3' end of that strand, and a new nucleotide is being added. The nucleotide is a triphosphate. Having 3 phosphate groups allows this polymerization reaction to be exergonic and spontaneous. Notice that the next nucleotide to be added is thymine. This is necessary because thymine is complementary to the existing adenine, so only thymine will fit properly. This is what we mean when we say that one strand serves as the template for the other. The strand that's being made is being made with an exact sequence that depends on the original strand. However, note that the new strand is not the same. Rather, it's complementary. By knowing the sequence of one strand, you absolutely know the sequence of the other strand. You can check yourself to figure out what base would be added next. Okay, let's take a step out and look what happens when DNA is synthesized. One of the most important things that happens is that the double helix must unwind. Once it's unwound, enzymes are able to access the individual strands and use them as templates for creating a new strand. There is, however, one important complication, and that revolves around the fact that DNA can only extend in the 3' direction. When DNA is synthesized, the two strands are split apart by enzymes. This is called the replication fork, and as the enzymes move along the double helix, the replication fork also moves along the double helix. Replication along the two strands is different because of the directionality of DNA. On the upper strand shown here, the 3' end that's being synthesized is advancing into the replication fork. Therefore, as the replication fork advances to the right, the new strand is assembled simply following that advancing replication fork. This strand is called the leading strand because it's synthesized in a continuous manner, and the synthesis is always following just behind the enzymes that unwind and divide the original DNA. Replication along the other strand is more complex. Since DNA is only replicated at the 3' end, the replication is directed away from the advancing replication fork. Therefore, as the replication fork advances to the right, the replication on that strand advances to the left, leaving an unreplicated region between where the new strand is synthesized and the replication fork. Here, you can see that the replication fork has advanced to the right, and the leading strand synthesis has followed directly behind it. The lagging strand, however, has advanced in the opposite direction, leaving a region of single-stranded DNA that has yet to be replicated. Eventually, this region will have an additional strand synthesized, again in the 3' direction, away from the replication fork. These small bits of newly synthesized DNA are called Okazaki fragments. In bacteria, replication always starts at a particular place on the circular chromosome called the origin of replication. Proteins help to split the two strands of DNA apart, and once apart, enzymes synthesize the new strands of DNA in the 5' to 3' direction. As this progresses, the replication bubble expands and replication continues on both strands, with the leading strand directly following the replication fork and the lagging strand synthesizing DNA in Okazaki fragments. In bacteria, this continues until the whole chromosome has been replicated. Eukaryotes, however, have hundreds or thousands of times as much DNA as prokaryotes, and synthesizing it in this way would take a long time. Instead, eukaryotes use multiple origins of replication along the chromosome. As each replication bubble is opened up, DNA is synthesized along both strands until the replication bubbles eventually fuse with one another, resulting in fully duplicated chromosomes. Okay, now, not surprisingly, this is a very complicated process, and there are many enzymes involved, each of which with a very specific job to do. We're going to concentrate on a small handful of those enzymes. The first is helicase, which opens up the double helix. Remember that when the DNA is in its double helix form, it's relatively inert, and other enzymes can't access it. Helicase does its job of opening the helix basically by breaking the hydrogen bonds between the two strands. Once the DNA strands have been separated, they have a strong affinity for each other and would like to find their way back to each other and reform their hydrogen bonds. In order to prevent this, cells use single-strand binding proteins, or SSBs, that hold the two strands apart until the new strands can be synthesized. Now that the helicase advances along the double helix, it pushes the twist in the helix further and further down. This can result in supercoiling the DNA, similarly to separating two strands of twisted rope. If you simply pull the strands of rope apart, the twists move further down and eventually bind up, causing supercoiling. The enzyme topoisomerase briefly cuts one strand or the other in order to spin one strand relative to the other, relieving the twisting strain. As we'll see momentarily, the main enzyme that synthesizes DNA is called polymerase. However, polymerase can't work by just joining onto a single strand of DNA. Instead, it needs to bind to existing double-stranded DNA, and from there it can synthesize new DNA at the 3' end. In order to supply this second strand onto which it can bind, there's an enzyme called primase, which synthesizes small sections of RNA that is complementary to random pieces of DNA. Because it's used to prime the DNA polymerase, this small section is called a primer. Notice that this primer is made out of RNA, not DNA. This will be important later. Once the primer is in place, the DNA polymerase can bind and start its job of assembling nucleotides into a new strand. In prokaryotes, this is done specifically by DNA polymerase III. In eukaryotes, there are several more polymerases, but the job is essentially the same. Finally, the sliding clamp helps keep the DNA polymerase in place along the template strand. Now, this has been happening on the leading strand, and so all that needs to happen is for the DNA polymerase to keep following the replication fork. On the lagging strand, the process is largely the same with a few modifications. As before, helicase separates the strands, and the single-stranded binding proteins keep them separate. Primase lays down a small RNA primer, and DNA polymerase binds to the 3' end of the primer and begins to synthesize new DNA in the 3' direction, away from the replication fork. Eventually, the DNA polymerase III will encounter the 5' end of another Okazaki fragment that was laid down previously. At this point, the polymerase dissociates from the DNA strand. After the DNA polymerase III dissociates, DNA polymerase I attaches and rides along the double-stranded DNA until it finds an RNA primer. The polymerase then gets rid of the RNA nucleotides and replaces them with DNA nucleotides. Once DNA polymerase I reaches the 3' end of the primer, it encounters the 5' end of the previous Okazaki fragment. The polymerase is unable to attach the 3' end to the sugar phosphate backbone that it's been assembling to the existing one in the old Okazaki fragment. Therefore, there remains a small nick in the backbone. It is the job of DNA ligase to find these nicks and seal them, eventually making the DNA strand have a continuous sugar phosphate backbone. Up until now, we've been looking at the proteins and enzymes associated with DNA replication as if they're all in a line along the DNA strand. In fact, it seems that many of these enzymes and proteins form a replication complex that's all bound up together and keeps the replication in one location. It may even be the case that rather than the enzymes moving along the DNA sort of feeds into a stationary replication complex. There's a really cool animation that shows this, and I encourage you to check it out. You can click on it in the link on the top right corner. As the polymerase moves along the template strand, it adds nucleotides that are complementary to the template. However, occasionally it makes a mistake and adds a base that's not complementary. When this happens, many polymerases have proofreading capabilities and are able to detect the mistake, get rid of the mistake, and then replace it with a properly matched nucleotide. However, even this sometimes fails and the wrong bases get added. Shortly after synthesis, other enzymes check the new strand for mismatches and conduct mismatch repair. The enzymes identify mismatches by the distortion they cause in the double helix. They can identify which is the original strand and which strand has the mutation because in the older strand, molecular markers called methyl groups are present. Therefore, mismatch repair systems only work during the time between when the new strand is synthesized and when those methyl groups get added. After that time, it isn't possible for the enzymes to identify which is the original and which is the new mutant strand. Another type of repair is called nucleotide excision repair. In this type of repair, newly synthesized as well as old, damaged DNA can be repaired. Damaged DNA is distinguished from good DNA by distortion to the double helix caused by either mismatched nucleotides or, in this case here, thymine dimers, which bind to each other along the same strand rather than pairing across to the other complementary strand. Thymine dimers are often caused by ultraviolet radiation on the skin, and people with mutations that make their repair enzymes non-functional are very susceptible to skin cancer. In order to repair these damaged sections, nuclease enzymes are used to excise the damaged DNA, then DNA polymerase lays down new nucleotides, and finally DNA ligase seals up any nicks in the sugar phosphate backbone. As a result of all these repair mechanisms, DNA can be faithfully copied from our original fertilized egg many, many times as our cells proliferate and get replaced throughout our lifetimes. There's one last difficulty that we need to address, and this only pertains to linear chromosomes. When the replication complex reaches the end of a chromosome, called the telomere, some parts of the complex can do their job, but other parts can't. Here we see the DNA being unwound completely all the way to the end. And the synthesis of the leading strand can easily complete all the way to the end. Shortly after the leading strand completes its synthesis, the lagging strand is almost complete. The last Okazaki fragments are joined together. However, there isn't enough space at the end of the chromosome to insert a primer for a new Okazaki fragment. As a result, a small portion of the chromosome cannot be replicated on that side. The same thing would happen to the 3' end at the, of the other strand. Now, this doesn't seem like a big deal, but after many, many replications, the ends simply get shorter and shorter. As a result, it's important that telomeres exist to prevent the chromosome shortening so much that they shorten into functional genes. Surprisingly, the shortening of telomeres is actually beneficial in some ways. We think that telomeres have an important role to play in reducing the number of replications that a cell can make. This helps to prevent uncontrolled growth, as in cancer cells. Eventually, the shortened telomeres will start shortening into functional genes, and the cell will become unhealthy and hopefully undergo apoptosis. You may think that if telomeres only ever get shorter, then our offspring will have shorter telomeres than we do, and their offspring will have even shorter ones. Therefore, over time, our chromosomes would always be shortening, and this would eventually impact genes near the end of the chromosomes. In our sex cells, however, we have an enzyme called telomerase that becomes active. Telomerase has a built-in RNA template and can add nucleotides to the telomere that are complementary to its own template. It does this a number of times until the template strand is long enough to support its own RNA primer, and DNA polymerase to extend the primer as it does with normal DNA replication. Therefore, our sex cells have a mechanism for extending the telomeres, duplicating without constantly losing telomere DNA. Unfortunately, cancer cells often have mutations in the regulatory sequences that control the expression, the transcription and translation of the protein telomerase. As a result, telomerase is often found to be active in cancer cells and is one of the reasons why cancer cells become essentially immortal. Whereas most cells have a limited lifespan due to the length of their telomeres, cancer cells are able to maintain their telomeres and therefore replicate indefinitely.